Welcome back. These are the week 3 slides for Bio5 about how cells communicate. So I just wanted to point out a couple of things before we get into the actual presentation. First is that there's going to be a couple of spots where the slides are just slightly different from the ones that are posted to Canvas. The first one is this slide where the title of the slide is a little bit different. Instead of saying cells, organs, and skeletal system, it says how cells communicate. So I'm just kind of integrating the ideas together and streamlining it based on what we're actually going to cover. Originally, when I set out the schedule, I thought that it would make sense to start talking about the skeletal system this week, so kind of cut back on what we have to do next week. But I think it'll make a little bit more sense to talk first about how cells communicate, keep it a little bit simple in that way, and then talk about muscles and the skeletal system together next week. So that's going to be a little bit different from how it was originally set out to be. When we talk about passive diffusion or simple diffusion, there's also going to be a spot where the slides are a little bit moved around, and I'll point out where that is so that you can keep an eye out for that when you download actual slides. The other thing that I wanted to point out is that when you're watching this, the video may or may not be captioned. YouTube auto captions, but because these presentations are a little bit longer and because there's kind of longer terms in the presentation, it might take a little bit of time to auto caption. So just be patient. It will happen within about 24 hours of posting. Okay, so thinking about how these chapters tie together, we're talking about chapter 5, chapter 9, and chapter 33. So we're building off of some ideas from last week, which we'll get to in just a moment. But chapter 5 is thinking about the cell membrane. So that cell membrane is very important for transport in and out of the cell. That cell membrane also receives signals from itself or from other cells. And then those cells are able to coordinate function. They're able to work together to form tissue and then eventually organs. So these chapters 5, 9, and 33 are thinking about cell signaling, about stuff getting in and out of cells, and about coordination of function. Thinking about last week, you should be keeping in mind characteristics of eukaryotic cells. Remember that this is a human biology course, so even though last week we talked about prokaryotes and kind of diversity of life, we're going to be focusing a lot on human cells and animal cells moving forward. You should also think about cytoplasm, what that actually is. I'll review that in just a moment also. And then this idea, again, that human cells don't exist individually most of the time. Some of the exceptions to that are gametes, so things like eggs and sperm. But in general, we have tissue bound together that's coordinating its function. We're going to start out by talking about chapter 5, which is the structure and function of plasma membranes. Before we get into new material, though, I wanted to review some slides that you saw last week so that they're fresh in your mind and that you can continue to build off of that information. So when you were going through these different sections, we're going to be talking about section 1, 2, 3, and 4. First, thinking about the components and the structure of the plasma membrane. Last week, I mentioned this idea of a fluid mosaic model, so you should keep in mind what we actually mean by fluid and mosaic. 2 through 4, we're thinking about passive, active, and bulk transport. So you should keep in mind whether these types of transport have a cell energy cost, whether they require the cell to spend ATP. Okay, so these slides are from last week. So remember that there are some kind of differences in structure between prokaryotic and eukaryotic cells, but eukaryotic cells do have this really beautiful, elegant fluid mosaic membrane. So we're going to talk in just a moment about components of that membrane, the phospholipid bilayer, and all of those proteins that are kind of embedded in that layer. Remember also that inside the cell, there's a lot of really important material that's distinct from outside the cell. So we have cytosol, which is a fluid medium And so that cytosol has a very distinct set of electrolytes. It has a very specific ion concentration in it that establishes a concentration gradient between the inside of the cell and the outside of the cell. That terminology will make a little bit more sense moving forward when we talk about diffusion. There's also organelles and the cytoskeleton, and all of that comes together to make the cytoplasm. And again, remember that human cells don't exist individually. They're working together, they're coordinating function as tissue, and in order to do that, they have to be able to connect to one another physically and also send signals. So this was plant cells have plasmodesmata. Animal cells have these gap junctions where you are able to send ions and other different things between cells. So they're a gap, they're open, they're a junction, they're a connection between cells, but they're an open connection. We contrast that with tight junctions, which just connect the cells together and don't allow any material through. And then there's also the desmosomes, which are similar to tight junctions, but they allow a little bit more stretching and flexibility. Okay, so getting into new material and thinking about what that phospholipid bilayer actually consists of. So phospholipids are unique in that they're amphiphilic molecules. So let's break that word down for just a moment. If you think about amph, like ambidextrous or an amphibian, something that's ambidextrous or someone who's ambidextrous, that AM prefix means that they're able to use both of their hands equally well. An amphibian lives in both water and land. They're not obligate one or the other. And so amph tells you that this molecule has two different properties to it. And philic tells you that it likes two different properties. So amphiphilic molecules are both hydrophilic, they're polar, and they're also hydrophobic, they're nonpolar. So it just depends on the part of the molecule you're talking about. That phosphate head is hydrophilic, those fatty acid tails are hydrophobic. So if you've ever used micellar water or micellar cleansing wipes, these are based on the same amphiphilic property. The fatty acid tails are going to connect with makeup that's on your skin that's kind of greasy, or just oil or grease on your skin. And then the hydrophilic part, the phosphate head, is going to connect to the water in the solution or water when you rinse off. So this is really useful for removing makeup and cleaning your face, and it's based on the same chemical property. Micelles are formed when you have these amphiphilic molecules. They form these cute little circles called micelles. These sort of self-aggregate in that way. So that single layer lipid sphere down in the bottom right, that one is a micelle. We see the lipid bilayer kind of aggregating in the similar way in the cell, but these micelles naturally form whenever you have these amphiphilic molecules. So amphiphilic molecules arrange themselves into micelles or liposomes. So in general, most of the cell membrane is that phospholipid bilayer. So phospholipid is that amphiphilic molecule, the phosphate and the lipids, the fatty acids. Bilayer is telling you that there's two layers. So the like components like to stick together. So the fatty ones, the fatty acids that are hydrophobic kind of stick together, and they protect themselves from the aqueous environment of the intracellular fluid and the extracellular fluid. So they want to kind of hide from that water-loving environment inside the cell and also outside the cell. So most of the bilayer is that, or most of the cell membrane is that phospholipid bilayer. One thing I want to point out is I've used a couple of different terms. So we have phospholipid bilayer, which is talking about the actual material. We have cell membrane. We also call it plasma membrane. So all of those are meaning the same thing. The phospholipid bilayer is the most narrow definition though, because that's specifically talking about those phosphate and fatty acid molecules forming this bilayer. But the whole plasma membrane, the whole cell membrane, has a lot of other stuff going on. It's this fluid mosaic structure. So first thing that I want to point out is kind of along the outer surface. So the top part of this diagram is kind of showing how the cell looks on the outside. The bottom part is showing the internal environment of the cell. So the outer surface of the cell has this glycocalyx, which includes both glycoprotein and glycolipid. I'll get back to that in just a moment, but I wanted to point out that I added kind of a note there. We have these integral membrane proteins that are integrated into the membrane. So they're proteins that are embedded in the membrane. We have channel proteins that stretch across the membrane and form these channels, and they selectively allow material through. So we'll talk about that when we talk about diffusion. We have these glycoproteins and glycolipids that are part of the glycocalyx and are involved in cell recognition. So they're along kind of the outer surface of the cell. They're going to be receiving signals from other cells, which we'll talk about later. And we also have these peripheral membrane proteins. So peripheral means kind of along one side. And so these are not stretching across the whole membrane, they're just integrated into part of the membrane. So they're peripheral membrane proteins. So we have a lot of different structures in this fluid mosaic model. So that's the mosaic part, but we also have a very fluid structure here. So if we look at different types of fatty acids, you've probably heard of saturated fats and unsaturated fats. So those terms have to do with the carbon skeleton and whether there's double bonds or single bonds. So if there's only single bonds, if it's what we call an alkane carbon structure, then we say that it's saturated. It's got the maximum number of hydrogens, it's fully saturated. But if it has double bonds, then it's unsaturated. It doesn't have the maximum number of hydrogens, and it also forms these kind of kink structures where it's bent, it's not completely flat. So if you think about fatty acids being pressed together, if they are saturated, you're able to get a lot of them pressed together, very rigid in structure. If they're unsaturated, they kind of maintain space between them. They're a little bit more fluid. So just keep that in mind. So when we're thinking about those structures, whether it's saturated or unsaturated fatty acids, that's when we're getting into fluidity. So those saturated fatty acids, when you're in a very hot environment, they keep things very stable. If you're in a very cold environment, stuff is starting to get kind of frozen in place, not able to move as much. You're losing kinetic energy. In that situation, you want unsaturated fatty acids in your membrane because that will keep things kind of loose and flowing. So we often talk about lipids in the context of triglycerides, phospholipids, fatty acids, but there's a lot of other fat-like structures, and one of those is cholesterol, which is a really important part of our membrane. So remember that there's a lot of molecules where people just think of them as being all bad. Cholesterol is really important for your cell function, and it's important for buffering both in hot environments and cold environments. This video, I would recommend opening up and going through on your own time. It's a really cool video that allows you to kind of explore the fluid mosaic model of the plasma membrane. Okay, I thought that I could maybe go back and show you part of it, but I'm not sure if it will allow me to record, so we're not going to stress about that. So thinking about kind of stuff moving through the plasma membrane, I mentioned that those protein channels are kind of selective. So we call the membrane selectively permeable or semi-permeable because based on different properties of molecules, they may or may not be able to easily cross through the plasma membrane. So when we have very large molecules, those can't easily squeeze through gaps in the membrane. They can't easily squeeze through even those protein channels. And then polar molecules have a hard time because the internal environment of the plasma membrane is very nonpolar, very hydrophobic. Material has to be or has to move through material that's similar to it. We say that like dissolves like. So if we have polar molecules, those would not be able to pass through the nonpolar environment of the plasma membrane. So we have to have other means of getting across. So when we're thinking about how stuff gets across this fluid mosaic membrane, there's a few different ways of kind of breaking this down. There's diffusion, there's active transport, and then there's this bulk transport. So simple diffusion and facilitated diffusion are going to be passive transport. Active transport and co-transport are both active transport, and then endocytosis and exocytosis are what we call bulk transport. So we'll talk first about simple diffusion and facilitated diffusion. And before we get into that, I want to kind of explain what we actually mean by ATP. So ATP stands for adenosine triphosphate. I'm not going to ever quiz you on that term on an exam. I'm not going to expect you to know adenosine triphosphate versus tetraphosphate or something like that. But the number of phosphates is important because popping off one of those phosphate groups is how we access energy. So one way of thinking about this is ATP is kind of like the currency of the cell. It allows us to move energy around and store it and use it. This ATP is produced by cellular respiration. If you remember terms like glycolysis or the electron transport chain, when cells undergo cellular respiration, they produce a lot of ATP that we use for cellular processes. So that ATP ends up getting broken down into ADP and then an inorganic phosphate. I'll show you an image that shows that a little bit better in just a moment. But remember that energy is stored in chemical bonds. And so when we have a bond in place that represents stored energy, if we break that bond and pop off one of those phosphates, we're releasing energy. And because these are covalent bonds, they're very strong. So we're able to access a lot of energy pretty quickly. So I think this image does a really great job of showing that. You can think about ATP like a charged battery or like a very full wallet. It represents a lot of potential that's stored in place. And then when you open it up or you use it in some way, you're going to kind of drain that molecule, you're going to take out the money, and then you go through these processes again to put that money back in or recharge that battery. So the cycle between ATP and ADP is how we store and use energy in our cells. And that all comes down to breaking that covalent bond to release energy for cellular processes. So when we're thinking about energy cost, there's a few different synonyms we use. If there's no ATP used whatsoever, just based on kind of, normal chemical processes, if it's just kind of the way things happen, you don't have to spend any energy, we call that passive. So a passive process means that no ATP is used by the cell, there is no energy cost. This is tricky because when ATP is used, when there is an energy cost, we say that it's active. So you would kind of think that all cell processes are active. There's a lot of dynamic stuff happening, but these are very technical terms. Passive specifically means no ATP. Active means ATP is used. So in thinking about diffusion first, this is a passive process. And so I'm hopeful that this recording is going to show you the GIF that's going on in the bottom right. But what it's showing is it has this bottle, it's full of green little circles that are tiny and can fit through the bottle. And as the bottle opens up, they kind of leave the bottle and equilibrate. The concentration of them inside the bottle and outside the bottle eventually stabilizes. So when the bottle first opens, it has what's called a strong concentration gradient. There's a very high concentration of this green molecule inside of the bottle and a very low concentration outside. So stuff moves along its own concentration gradient. It goes from where there's a lot of the substance to where there's not as much substance. You can think about this like putting a drop of food coloring into water. It starts out very strong in one particular location and then it spreads out. Or if you spray perfume on one side of the room, it starts out very strong on that side, but then eventually diffuses out throughout the room. So that's the idea of diffusion. Keep in mind that there are limitations here though. So those blue circles that are around the outside container are too big to get in. So they have their own strong concentration gradient, but they can't really do anything about it. They can't move in response to their concentration gradient because there's a size limit. They can't get into the bottle. So substances diffuse from where there's a lot of them to where there's not as much of them without spending any energy. It's a passive process. It's just the way stuff moves. So here we have this happening in and out of a bottle. We gave the example of being in food coloring in water or perfume across a room, but oftentimes, especially in the cellular environment, we're talking about concentration gradients across a membrane. So diffusion is happening from one side of the membrane to the other. So this image is showing that we have the extracellular fluid on top, we have the plasma membrane, that phospholipid bilayer, and then we have the cytoplasm on the inside. On the left side of this image, we have a very strong concentration gradient. Over time, molecules are going to be able to diffuse through the membrane, make their way into the cytoplasm, and eventually equilibrate between the extracellular environment and the inside, the cytoplasm. So here we're looking kind of at the concentration and keeping in mind that this looks like a pretty small molecule. It's probably not charged, it's probably non-polar, so it's able to make its way quite easily through the membrane. So that is kind of looking between the extracellular environment and the intracellular environment, but the movement doesn't just happen in one way. It depends on the concentration gradient. So in this image, we have a very high concentration on the left side of the red molecule inside the cell, and it's going to end up moving outside the cell. In part B, we have the opposite concentration gradient. We have a very strong concentration outside and very little inside. So the net movement is going to be from the outside inward. So this is a really important point to keep in mind, that whenever we're talking about a concentration gradient, we have to think about relative terms. We're not just saying that something is a very strong concentration gradient on its own, we're comparing it to something else. And in just a moment, we're going to get terms to kind of classify that relationship. So substances can leave or enter a cell. They can move across the membrane depending on the concentration gradient that's established. Okay, so before we get further, I wanted to kind of review some terms that you maybe learned in chemistry and maybe haven't learned, but they are important to kind of think about. So when we're saying the term solute, that is a substance that is dissolved in another substance. So for example, if this cup is showing salt water, that's the salt, that's the NaCl. The solvent is the substance in which the solute is dissolved. So it's doing the dissolving. It's the dominant characteristics. One example that I sometimes give for this is when you put gummy bears into vodka. The gummy bears and the dye in them are the solute. The vodka is the solvent. It ends up still looking pretty much like vodka at the end of it. And so for salt water, it still basically looks like water. It just has solutes dissolved in it. So when we have that process finalized, we have what's called a solution. It's homogeneous. There's only one phase to it. It's not chunky. It's a smooth mixture of the solute and the solvent. So we might have one solution with 1 degree of saltiness and another solution with another degree of saltiness. One might be more dilute than the other. So we're going to start comparing the amount of solutes or the concentration of solutes in one solution versus another. Those different solutions are usually on either side of a plasma membrane. So when we're thinking about movement, when we're talking about the solutes moving, that's called diffusion. If the solvent moves along its own concentration gradient, that's osmosis. And so the movement is going to be predicted based on the tonicity of the solution. So when we use the term hypertonic, hyper should make you think about like hyperactive, hyperglycemic. That means more. So you have a lot of something. So a hypertonic solution has more solutes relative to another solution. Hypo maybe makes you think about hypothermia, really low temperatures. Hypo means less, or hypodermic needle. So a hypotonic solution has less solutes than another solution. Iso maybe makes you think about isosceles triangles where they have sides of the same length. Iso means the same. So isotonic solutions are the same concentration on either side of the membrane. So let's go ahead and kind of look at these few different examples. In this first one on the far left, A is hypotonic relative to B. So let's say that A is the outside of the cell, B is the inside of the cell, and we have a cell membrane in the middle separating them out. So which way will the solutes move? So hopefully you predicted that they would move up into A. So they're moving from where there's a lot of them to where there's not as many of them. In this middle example, A and B are isotonic. And so in that situation, it's a little bit tricky. They're actually going to be moving back and forth. So when we're talking about diffusion, we're talking about net movement. We're thinking about overall what is happening. And so here, some solutes from A might trickle into B, some from B might go into A, but overall they even out. In this last example, A is hypertonic relative to B, so the solutes should move in a net direction towards B. So when thinking about what factors affect this, we think about the strength of the concentration gradient. If it's very, very concentrated on one side and not as concentrated on the other, there's going to be a strong diffusion from more concentrated to less concentrated. If the molecules are tiny, they're probably better able to pass through a membrane. The hotter it is, within reason, the faster diffusion happens. That's why it's easier to dissolve sugar or salt or tea into hot water than it is into cold water. Also, distance traveled. If you have a very high surface area to volume ratio, then diffusion is easy. If it's a low surface area to volume ratio, it's harder to diffuse. And there's also different qualities of the plasma membrane, the solutes and the solvents themselves, whether they're polar, nonpolar, charged, different things like that. So I mentioned osmosis. Remember that water is moving along its own concentration gradient. It moves from where there's a lot of water to where there's not as much water. So water can do work. It can actually defy gravity. It goes to where there's less water in the solution, which means maybe more solutes. So we can also say water follows salt, or water likes to dilute things. It goes to where there's a lot of solutes. So this is where the slides get kind of a little bit out of order, but I wanted to introduce osmosis before I talk to you about animal cells and plant cells. So what is the main difference between animal cells and plant cells that we always hear about growing up? Hopefully you're aware that plant cells have a cell wall, animal cells do not. And so there's going to be some differences in structure and also functional response accordingly based on that difference. So here we have red blood cells. Those are animal cells, they're our cells, they don't have a cell wall. So when those red blood cells are in a hypertonic environment, when they're in a very salty environment, water is going to leave the cell in order to dilute the outside environment. So those cells are going to shrivel. When it's isotonic, when you have an environment outside the red blood cells that's the same as inside the red blood cells, water is going to leave the cell, but it's also going to come back in, and that's a very healthy environment. For a hypotonic solution, water is going to keep entering the cell. So this is, for example, if you have pure water and the red blood cells are inside of that pure water, water is going to keep entering the cell to try to dilute that internal environment of the cell, and it's going to keep doing that until the cell eventually swells and bursts. So this is kind of like filling up a water balloon. If you just keep filling it and filling it, eventually it will burst. So this is why it's really important when you guys are nurses not to ever inject a patient with pure water. You have to use saline solutions or different solutes within those IV drips. So plant cells are different. They have a cell wall in addition to a cell membrane. And so when they're in a hypertonic condition, yes, water is going to leave the cell. It's not a great situation. Below that, on the far left, you can see the outline of the cell wall. You can see the chloroplasts all kind of condensed together because the membrane has shriveled up inside of the cell wall. In an isotonic condition, we still have water coming into the cell and leaving the cell. The chloroplasts are a lot happier. They're kind of floating around, more spread out. That's an ideal situation. And even better, we have a hypotonic condition. So you don't have to prepare a saline drip when you water your plants. You just use normal tap water or pure water. And the reason for that is that pure water is totally fine to get into those plant cells. They can keep taking on water. They have a vacuole to store it. And then they also have a cell wall to reinforce it. So the cell is not going to be able to swell and burst because it has that cell wall protecting it. So here, when a plant cell is in a hypertonic solution, when it has net water loss, it's going to wilt. But when they are in isotonic or hypotonic solutions, they're taking on water, they're able to stay upright. So we see not just a cell response, but a whole organism response as well. So that was simple diffusion, but diffusion can also be facilitated to allow for kind of small molecules to pass through that membrane. So it's still not requiring any energy, it's still a passive process, but when we have transport proteins like channel proteins and carrier proteins, those are going to allow material through the membrane without costing any energy. So next we're going to get into active transport and co-transport, which is also called secondary active transport. So remember, when we say active, we mean that this costs energy, that the cell is going to spend ATP to carry out a process. So active transport is really important for dealing with those factors that make diffusion difficult. So there's a few of those. Remember, we talked about what makes diffusion easy, but in certain situations, we can't easily carry out diffusion. So in this example, we have a negative charge inside the cell and a positive charge outside the cell. So we have kind of a difference in charge there. For potassium ion, that's K+, it is very strongly concentrated inside of the cell. So there's a concentration gradient there. You would think that it would be able to passively flow outside of the cell. But the problem is that potassium is positively charged, and there is a positive charge already existing outside the cell. So it's kind of like trying to put two ends of a magnet together. So like the two positive ends of a magnet together, they're going to repel each other. So this concentration gradient is working opposite of the charge differential. So in this situation, we would need to use active transport in order to move those potassium ions outside of the cell. So one example of active transport that's really important is the sodium-potassium pump. So whenever you see that Na plus K plus ATPase, or you hear sodium-potassium pump, that's talking about a very specific type of pump that carries out active transport across a membrane. It's really important for your cells. Your cells end up spending tons of ATP on these sodium-potassium pumps. So what's happening here, like we saw on that image on the previous slide, sodium is higher in concentration outside the cell, potassium is higher in concentration inside the cell, and both of them have these concentration gradients. So you have, in this situation, 3 sodium ions and 1 ATP binding to the pump. ATP is going to be split, it's going to release one of those phosphate groups, and sodium is going to get pumped outside the cell to kind of maintain that concentration gradient. And then sodium is released, the enzyme changes shape, and then potassium binds and is transported inside the cell. So you have maybe trying to maintain that concentration gradient. You have sodium being moved outside the cell, you have potassium being moved inside the cell, and you're using ATP to accomplish that. So it turns out that about 30% of ATP in most cells is actually spent on these sodium-potassium pumps. 70% of ATP in nerve cells is spent on these sodium-potassium pumps. When we talk about the nervous system, we'll kind of understand that a little bit further, but it comes down to this idea of action potentials. So just keep that idea in mind, keep the idea of electrochemical signaling in mind when we talk about nerve cells. So we talked about generally active transport. That active transport might be primary or it could be secondary. So when it's primary, that means that the ATP is directly being used for that movement. So primary active transport directly uses ATP to move solutes against their concentration gradient. So here we have this example of sodium ion being pumped outward using ATP, and then we have this concentration gradient being established. Then if sodium ion were to move into the cell and also carry in another molecule, we would say that that other molecule was being moved in through secondary active transport. It relied on the ATP building up a concentration gradient that is then used to carry that other molecule. So secondary active transport uses the electrochemical gradient that's established by primary active transport. Both of these use ATP, it's just a matter of whether that ATP is directly being used to move the molecule or the ion. So the final type of transport that we're going to talk about is this bulk transport, which comes down to endocytosis and exocytosis. So when we're thinking about that term endocytosis, endo is thinking about inside, so the word endo means inside, cyto is cell, and cis is kind of like movement or a process. So endocytosis is the movement of material into a cell. So it's how large molecules enter a cell. And so when we're thinking about this, we can kind of divide it up into phagocytosis and pinocytosis. Phagocytosis is how cells take in very large particles, and this involves pinching off of the plasma membrane to form a vacuole. So we have what's called invagination, or kind of the cavity forming along the outside portion of the plasma membrane that surrounds that large particle. It forms a vacuole and it kind of pinches off from the rest of the plasma membrane, which heals itself, kind of sticks back together, and now you have this large particle inside of the cell. For pinocytosis, this looks, or like it kind of appears like the cell is just taking in extracellular fluid, but what's actually happening is that there's small molecules or ions dissolved in that extracellular fluid that is being taken in bulk from the outside environment into the cell. So this process of endocytosis is active. It requires energy, as is exocytosis. Exo means outside, cyto is cell, cis is a process. So we have the movement of material outside of a cell. It's how material gets from inside the cell to outside the cell. So we have the reverse of endocytosis. We have vesicle formation inside of the cell. Those vesicles move towards the plasma membrane, they reintegrate with the plasma membrane and release their contents into the extracellular environment. So again, this bulk transport requires energy. It costs ATP. Okay, so we're going to briefly talk about Chapter 9, getting into cell communication. And we're only going to cover the first section, which is about signaling molecules and cellular receptors. So the rest of this chapter is going to be relevant when we talk about the nervous system. For now, we're just going to focus on what components are involved in cell signaling and basic types of cell signaling. Okay, so remember that there's a lot of structures on that fluid mosaic model, on that cell membrane, that are specifically involved in cell signaling. So as that glycocalyx, we have those glycoproteins and we have glycolipids. So we have these protein and lipid anchors with carbohydrates floating on the outer surface of the cell membrane. Those are involved in cell-to-cell recognition. So an organism has to be able to identify itself as itself. It has to be able to say, okay, these are my cells, and it also has to be able to send signals between different types of cells or similar types of cells, or even within the same cell. So when we're thinking about those distinctions, there's words like autocrine, paracrine, endocrine along the right side. We'll get back to those in just a moment. But in general, when we have intercellular signaling, inter means between, so these are signals sent and received between different cells. We also have intracellular signaling, which is communication within a single cell. So when we have these signals being sent, it's not like the cells can just send a snap to each other. They have to send signaling molecules, and those signaling molecules have a specific structure-function relationship with receptors on another cell. So these have to match together perfectly. They're like puzzle pieces. Not every cell has the receptor for a particular molecule, so it's a very structure-function relationship. So a signaling molecule is sent out. It can be sent out to a cell very close to it, or to a cell on the complete opposite end of the body in a completely different system, but somehow that signaling molecule makes its way to that receptor. It binds to the receptor, and then something happens. So we'll talk about what happens. So when we have that signal being sent out, another word for that signal is a ligand. And so we have the ligand being sent out by a particular cell. It binds to a receptor on the target cell, and you get a response. And again, there are going to be a lot of cells nearby that the ligand is going to pass by that do not have the receptor for that ligand. There's some situations where they maybe should have the receptor and there's a mutation and they don't, so then you're not getting a response that you need. But in general, it's good because you don't want every cell to respond to every signal. You just want the response to occur in target cells that are capable of dealing with that signal. So when that signal is received, we have what's called signal perception. So we have the ligand and the receptor binding, the cell perceives the signal, and then there's a chemical cascade. We have this intracellular signal transduction. So we have a series of reactions that occur that are kind of continuing because of that signal. We have a cascade effect, and then eventually we get the cellular response that we need. So all of these are kind of incremental changes. Maybe we have a molecule slightly changing in structure again and again until we finally get this response that we need. So I mentioned these words endocrine, autocrine, and paracrine. These are all different types of signaling. And so you might have heard of the endocrine system. Maybe you've heard about hormones. Those are signaling molecules. And so when we have endocrine signaling, this is happening across great distances in the body. So you could have a signal being sent out from your brain having an effect way on the opposite side of your body. You could have a signal being sent out from your adrenal glands or from your thyroid gland that's having an effect, again, completely on the opposite side of your body. So endocrine signaling is across great distances. If you think about the word auto, like an autobiography, that is having to do with the self. So autocrine signaling is signaling from a cell to itself. It's signaling within a single cell. And then paracrine signaling is signaling between close cells. So that prefix para means similar or at the same time as, or close to, and so paracrine signaling is signaling between neighboring cells. Okay, so the last chapter that we're going to talk about is chapter 33, and we're just going to talk about a specific part of it, thinking about the animal body, basic form and function, getting into primary tissue, and then so that's section 2. For section 3, I want to mention homeostasis is very important, but we're going to cover that section later in the semester. So today, all you need to focus on are those general types of tissue. So there's going to be 4 types of tissue and basic characteristics about them. There's not going to be a focus on the individual types of cells. I'm going to talk about them. You don't need to know them for the exam, but they are good to know kind of moving forward to prepare you for anatomy and physiology if those are classes that you do need to take. So I know some of you are taking this class kind of maybe to make up a grade or as a general ed class, but some of you are taking it in preparation for anatomy and physio, so I just want to make sure you're prepared for that, even though you won't be held accountable for it in this class. Okay, so the 4 main types of tissue, and this is something you should for sure know for the quiz, are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. And remember that tissue are these aggregates of cells that are kind of coordinating function in some way. So epithelial tissue, the purpose of them, their function, is to cover exterior surfaces, internal cavities, and passageways. So they're kind of this protective structure that's covering surfaces. Connective tissue connects that, it holds it together. So it binds together different cells and organs. It protects, supports, and integrates different parts of the body. Muscle and nervous tissue, you're probably a little bit more familiar with. Muscle tissue responds to signals. It responds to a stimulus, and it contracts and then expands or relaxes in order to move your bones around. So it's attached to bone, and it's able to kind of move our skeleton around. And I'm kind of dancing around as I'm saying this, and I realize you can't see me. But muscle is important for movement and responding to stimulus. Response can be voluntary or involuntary. We have control of our skeletal muscles, but there's a lot of muscle inside of our digestive system and inside of our circulatory system that we don't consciously control. There's also nervous tissue that is going to be sending signals, so propagating electrochemical signals to communicate throughout the body. So those are the 4 main types of tissue and their associated functions. This is a very important slide to know for the quiz and for the final exam. I'm going to work our way through each type of tissue, starting with epithelial tissue. So when we're talking about different types of epithelial tissue, we classify them based on the shape of the cell and also the number of layers. So for example, your skin has many layers of epithelial tissue or epithelial cells, and so it has a very particular name accordingly. So when we're thinking about the different shapes, if we say squamous, that means that those cells are very flat and thin. So if you look at the top part of that box, you can see that those cells are really flattened. They're not very tall, they're kind of extended outward, and when they're stacked on top of each other, they form very thin layers. Cuboidal, you should think about a cube. So these are as tall as they are wide. They're cube-shaped. And so they might be kind of in one layer. They might be stacked on each other, in which case they kind of get a little bit warped, maybe a little bit more circular. But in general, they're shaped like cubes or like squares. And then we have columnar. Those are like columns. They're very tall and thin. They're rectangular. And so we have different types of columnar epithelium. So the different columns that you're seeing here, simple versus stratified versus pseudostratified, have to do with the number of layers. So if we have simple squamous or simple cuboidal or simple columnar, it's just one layer. Stratified is 2 or more layers, and then pseudostratified is a little bit tricky. We see that a lot with columnar. It's just one layer, but the way they're kind of squished makes it look like their nuclei are kind of all over the place, and so it gives the impression of having multiple layers, even though it's just one layer. So it's a single layer that appears like multiple layers. There's also transitional epithelium, which kind of go from looking like columnar to looking like squamous as different organs kind of distend outward. You find that in your bladder, for example. So when the bladder is empty, it looks kind of like columnar epithelium, but then as the bladder stretches out, it starts to look more like squamous epithelium. So in that situation, that's called transitional epithelium. In lab, you should get the opportunity to look at some of these types of cells when you guys talk about cells specifically. Connective tissue is really important for supporting and protecting different organs and different structures within our body. This is stuff like fascia, tendons, your skeleton, blood, and lymph. And so when you look at all of those seem very different. Blood and connective tissue, or blood and and bone and cartilage all seem very different from one another, but they're all connective tissue because they all meet a couple of requirements. They have cells that are spread in some sort of ground substance. So you have the actual cells in something that's grounding them, like a fluid or a solid with protein fibers, and they all derive from the similar cell layer early on in development. So one thing that I want to point out that's kind of cool is that bone is living tissue. So I always think it looks kind of interesting. It looks kind of the way that like Van Gogh draws light, if you've ever seen Starry Night. So that is kind of how bone tissue looks. It's a type of supporting connective tissue, and it has many different types of living cells. It has osteoclasts, osteoblasts, osteogenic cells, and those are constantly building up and breaking apart this non-living matrix. So bone is a type of connective tissue. We also have muscle tissue. It's excitable, it's able to respond to stimuli, it contracts, it shortens, and generates a pulling force before then relaxing again. And there's 3 main types of muscle tissue. There's skeletal muscle, which is kind of a lot of the bulk muscle that we think about. We can control it quite easily, and so it's attached to bones and to skin. It's how we make facial expressions. And so we'll talk about skeletal muscle quite a bit next week. There's also cardiac muscle, which makes up our heart. And so this has really unique electrochemical properties, but it doesn't have voluntary control. So we can't control how our heart beats, but it is a muscle. It's a very strong muscle. And then there's smooth muscle, which is what we find in our digestive system. We also find it in our cardiovascular system. So this kind of keeps stuff flowing throughout our body, and we do not have control over it. We don't have voluntary control over it. We do have control over it, but it's involuntary. So finally, we have nervous tissue. So this is going to kind of help us process information about the world around us. It mediates perception and how we respond to it. And kind of the classical cell type that we think about when we think about nervous tissue are the neurons. They have a cell body called a soma, a nucleus inside of that cell body. They have these branches called dendrites that surround that cell body. A very long axon, which is actually wrapped in fat to help send signals, and then it has a synapse that connects with other cells. So the neuron is kind of the classical example of the cell type in nervous tissue, but there are a lot of other cell types as well. So we have what are called glial cells or neuroglia. These are support cells. These are things like astrocytes, microglia, oligodendrocytes, and Schwann cells. The type of cells are kind of different depending on whether we're talking about the central nervous system or the peripheral nervous system. I have information about each of those cells here. Again, it's just kind of background information to prepare you for anatomy and physio if you would like. We'll go into more detail about them later in the semester when we talk about the nervous system. Okay, so those are the chapters that we're covering today. Make sure that you look over the feedback from the last quiz. Again, I'm sorry about the glitch on the last question. Hopefully you still got to kind of see the correct answers. If you have any questions about those forms of cell connections, like the desmosomes and the gap junctions and tight junctions, please let me know. Make sure that you listen to the lecture, which hopefully you did since now you're at the end of it. The study guide will be posted tomorrow for the next quiz. If you have any questions at all, please email me. The online reflection will be posted pretty soon. Sorry for not doing that as yet, but it will be due Monday by midnight, and the quiz will be available between Monday and Wednesday.